Hello, I'm Dieter Steckless, and I want to welcome you to this second lab rotation, the Veterinary Sciences Lab. In this lab, we're going to focus on the mammalian heart. So I'm going to share my screen. Here we go. So here's what we're going to cover as part of this lecture. We're going to break this up into three parts. In the first part, we're going to cover the heart as part of the cardiovascular system. We're going to talk about the development or embryology of the heart, and very briefly about the evolutionary history of the mammalian heart. Then in the second video, we'll talk more specifically about the structure and function of the heart, including major vessels and how the blood flows through the heart and vessels. And then in the third video, we'll have a brief presentation of some comparative aspects of the heart, how it varies among different species of mammals, particularly in regards to heart size and rate. So let's get started in talking about the heart as part of the cardiovascular system. So when you see the terms cardio or cardium, it means that there's reference being made to the heart. It always implies something relating to the heart. When we think about the heart, we have to think about it as being a critical part of the cardiovascular system. And there are three components to the cardiovascular system. The heart, of course, is a key component, but then you also have blood vessels, not just ones emanating directly from the heart or coming into the heart, but also coursing throughout the body, supplying various tissues of the body. And then, of course, you also have the blood itself. And we're not going to be talking about the blood in this particular rotation. We just don't have time to do that. But let's just consider momentarily what the main functions are of the cardiovascular system. There are really three. One is transport. It transports nutrients, obviously, but it's also critical for gas exchange, particularly for taking in oxygen and getting rid of carbon dioxide. Also, thinking about the blood itself, it can carry an infection, but it can also carry parts of the immune system like antibodies that can protect you against infection. And of course, blood gets recycled very quickly. You produce new blood cells. So if there's blood loss, then you can replenish the blood. You have the capacity to replenish the blood. So there's also some protection against blood loss. Thirdly, it is important for maintaining the homeostasis of the organism, kind of balance, if you like. It's important for internal temperature regulation. Think also about the regulation of alkalinity or acidity that's in the tissues, what's known as pH. That's very, very important. And of course, fluid is very important for regulating fluid levels in the body itself. So those are the main functions. But let's turn here to a consideration of the development of the mammalian heart. It's embryology, as it's called. Now, the heart is one of the first organs to develop. Why is that? Well, because, as I say here, as the embryo grows, it can't rely on simple diffusion method, you might say, simple diffusion process for getting nutrients, for getting exchange of gases with different tissues, because the distance is basically too great, where the diffusion gradient is not sufficient. So you need to have a pump, basically, that can deliver, that can perform these functions for the growing embryo. And that's why it's one of the first organs to develop. Let's look a little bit more detail at the actual development of the heart. And let's focus on the right side of this image. We have an earlier stage in the embryo at the top and a somewhat later stage at the bottom. In the top, what I want you to note is that, well, I should say, first of all, this is sort of a cross section through the embryo, as though you were sort of cutting it in half. That is, in the middle between the tail end and the head end of the embryo. If you cut right through the middle of it, then this is the view that you would have of it. And what you can see here is that there are these heart tubes, one on each side. They're part of this interior of the embryonic plate, as it's called. This is the mesodermal part. You don't have to remember any of this, but just note that there are these two heart tubes, one on each side. And a little later in development, in embryonic development, what you see here is that these two tubes fuse together here in the middle. That's what you see here. These two tubes have now come together, they're fused here. And this is here now the development of the basic heart right here. Just this becomes a single tube, and this tube, as you'll see, becomes the heart. It's suspended here just below the gut. This is the developing gut here, labeled as one, just sort of suspended in this cavity here that later on becomes the thoracic cavity. Right? You can see it's already suspended there inside there. Here's another view of it. If we look at the embryo, here's the head end and here's the tail end. You can see that the heart develops at the head end, basically, just below the foregut. And here again, it shows these at day 20. This is the human embryo, by the way. You can see these two tubes, one on each side, that begin to fuse together. And then they begin to pump, they begin to pulsate. That's one of the characteristics of heart muscle. It naturally contracts in this way and it serves as a pumping mechanism, pushing blood around basically throughout the embryo, coming in one end and then out the other here. And so we have the development of the very primitive basic chambers of the heart. As we go along here, you can see here's at day 23 and going on all the way to 35, we're beginning to see essentially what looks like a four-chambered heart already, with the two ventricles and the two atria, the big aorta that carries blood away from the heart to the rest of the body. So very early on, by day 35, it's pretty remarkable. You have this four-chambered heart that's already pretty much taken form in the embryo. So here's something that's of evolutionary interest that kind of connects up with the next part of this presentation about the evolution of the mammalian heart, which is that we have evidence of this evolutionary ancestry in the form of what are known as these branchings of the dorsal aorta. This is the big artery that leaves the heart. That are called aortic arches. You can see they're actually numbered here. We have a series of these aortic arches that come off the dorsal aorta that's paired, one on each side. This is a remnant, an embryological remnant, actually really evolutionary remnant of having gill arches. That is, that each of these branches of the dorsal aorta runs through the gill system. As you'll see in fish, this is of course crucial for gas exchange. But in the case of mammals, and birds for that matter, you don't rely on gills for breathing. You have lungs. So these gill arches become transformed into other kinds of major vessels. For example, the big vessels then run up into the head and neck, like the carotid artery system here, one on each side. And the other aortic arches, these gill arches, if you like, basically go away. They get resorbed. And so then here, this is what you would see in the adult organism, the dorsal aorta coming off the heart and these major branches that are remnants of the gill arches, these primitive gill arches. So there's a stamp of evolution in the embryo here. Well, let's think more specifically about the evolution of the mammalian heart. It starts off, just as it does in the human embryo, as a single tube, basically, with blood coming in one end, coming in into here, the venous sinus, as it's labeled, into an atrium, into a single ventricle. These are the contracting chambers, the atrium and the ventricle, and they push blood out into the systemic circulation. So it's a very simple kind of two-chambered pump, atrium and a ventricle, single ventricle. Now, in fish, as I've mentioned before, you have gills. And so if you look at this single pumping mechanism here, an atrium and a ventricle, so blood comes in, it's oxygen poor and carbon dioxide rich, comes into the atrium, gets pushed into the ventricle contracts, and it pushes this blood into the gill capillaries. It pushes it into the gill arches, basically, and that's where it gets oxygenated in fish. There's no lung here. So this is how gas exchange happens in the fish. All you need is a pump, basically, a pump that can get the blood circulating throughout the fish's body. It doesn't have to be very complicated. By the time we get to amphibians and reptiles, we already see more chambers of the heart, additional chambers of the heart developing. We now are looking in the reptile, for example, at a right atrium and a left atrium, a right ventricle and a left ventricle. And we now have blood coming in to the right atrium, getting pushed out into a lung, because the reptiles were the first land mammals, and so they had evolved lungs to allow them to breathe on land, and this is where the gas exchange occurs. It's in the lung through the capillaries in the lung, so the blood gets pushed into the lung and gets, this is where gas exchange happens, and it comes back into the left atrium, and then it gets pushed out from there into the systemic circulation. So by the time you look at a reptile, you already have this four-chambered heart. Big difference is that the ventricles in particular are not all that well separated, that is the right from the left in the reptile, and they're not at all separated in amphibians either. But in amphibians, that's okay. It's okay to have some mixing here between the deoxygenated and oxygenated blood, because in part because, as you might know, amphibians have moist skin, and they can actually breathe, as it were, through their skin. They can achieve some oxygenation through the skin itself. And so the blood that comes into the right side of the heart is actually somewhat oxygenated. And so it's okay if there's some mixing that occurs there. That's it's basically not an inefficient heart. It does what it needs to do in an amphibian. And likewise in a reptile, reptiles are cold-blooded animals. They don't require high amounts of oxygenation for their tissues in the way that mammals and birds do. We'll come back to that. So in mammals and birds, you need to have much better separation of the ventricles, of the heart's chambers, as it were. So here's a mammalian heart now, and this is the same kind of heart we have. It's a fully four-chambered heart. It's a very efficient kind of two-pump mechanism, blood coming into the right side of the heart, right atrium, right ventricle, gets pushed into the lungs, and this is where you have oxygen exchange and carbon dioxide leaving the blood, comes back as oxygenated blood into the left atrium, and then it gets pushed out through the left ventricle into systemic circulation. Now you can see you've got very clear separation of these chambers, because you don't want any kind of mixing of oxygenated and deoxygenated blood. It's critical for mammals to have rich oxygen content in their blood. We'll come back to that a little bit later. So this concludes the first part of this lecture. We'll go to part 2 next, so stay tuned.